Is everyone still there? Sorry, I lost connection for a second. Looks like we still have everyone. Thank you for holding on and not leaving. I really appreciate that. Okay. All right, back to the phospholipid bilayer. So I was saying how important it is for you to understand its structure and its function in the cell. So we're going to talk some more about it. And so these details you will need to know. If my screen will work. Okay, so the plasma membrane. There is actually more to it than just phospholipids. So first of all, on the outside of the cell is extracellular fluid, or we call it interstitial fluid. And this is a water-based solution, a medium, that surrounds the cell and has important properties. The plasma membrane itself, or the cell membrane, separates the cytoplasm from the cell, inside the cell, from the extracellular fluid outside. So extracellular outside, and then cytoplasm on the inside. So this plasma membrane creates a separate environment on the inside of the cell compared to the outside of the cell. Okay, so functions of the plasma membrane. Isolation, so isolates the inside of the cell from the outside of the cell by forming a barrier. This phospholipid bilayer forms a very effective barrier that controls what is able to enter and exit the cell. So it regulates the exchange of substances with the environment. So it allows ions like calcium, sodium, potassium to enter the cell, and nutrients like glucose, and also allows wastes to be released and cellular products that have been created. So lipids in the membrane, you have your phospholipid bilayer, 2 layers. So just as a reminder, you have your hydrophilic heads, which means they are water loving. And that's great because that extracellular fluid is a water-based solution. So the hydrophilic heads that like water are facing towards the water. Then you have these hydrophobic tails that they're lipids, they repel water. So they're in the center of this bilayer, separated from the water. They form like the inside of your sandwich. So if the hydrophilic heads were the bread, the hydrophobic tails would be whatever you made your sandwich with. Okay, so having this hydrophobic inner core is great because it provides a barrier to ions and anything that dissolves in water. It cannot pass through the lipid because it repels. It also contains cholesterol, which I did mention before in the past, and cholesterol adds fluidity to the plasma membrane. It allows it to have more flexibility, for example. Oops. Okay, 2 structural classes of membrane proteins. You have integral proteins, and these are within the membrane. So in this picture here on the right, integral proteins would be embedded in the membrane, just like you see there. And so you would find them spanning the width of the entire plasma membrane. You can also call them transmembrane proteins, and they form things like channels that allow substances to pass through the plasma membrane. The other type of structural class of membrane proteins are peripheral proteins. And these are either bound to the inner or the outer surface of the membrane. So right here, is an example of a peripheral protein because it's on the inside of the plasma membrane. It does not extend through the whole plasma membrane. So in this picture here, you can see your integral proteins again, right here. You have a transmembrane protein right there. Okay, so functions of membrane proteins. One, anchoring proteins. And so these attach to the inside or the outside surface, which would make them integral proteins. If we back up, it would make them peripheral proteins. Okay. Then you have recognition proteins. And these recognition proteins, for example, right here, looks like a recognition protein, identifies the cell as being normal or abnormal. And so these proteins would be in, these proteins would be created inside the cell through protein synthesis and then transported through vesicles to the plasma membrane itself, where they are embedded in the plasma membrane and basically tell the other cells in the body if it's a normal cell or an abnormal cell. Then the third category of membrane proteins are enzymes. And if you remember, enzymes speed up chemical reactions. They reduce the activation energy of a chemical reaction, actually allow it to happen at a quicker pace than it would actually happen otherwise if it would happen at all. Okay, so more on these proteins, more classes. You have receptor proteins, and these bind to ligands, which examples would be ions or hormones. And when they bind to a ligand, or when ligand binds to it, it causes a change, a reaction. So it responds to them. Then you have carrier proteins, like this picture here, and they're very specific for a particular solute, like sodium, for example, or calcium. And they transport those specific ions or substances only into the cell. So you have specific carrier proteins for every different type of substance that's out there. You can have a carrier protein, or you can have a channel protein that's always open that allows certain substances to pass through. So for example, channel proteins regulate the flow of water and solutes through the cell. So it helps control that intracellular environment that's different from the extracellular environment and extracellular fluid. So the cell itself maintains a unique internal environment, and these proteins play a really important role in helping to maintain that internal environment of the cell. Just kind of like our bodies have internal regulations, for example, our temperature and our blood glucose levels. The cell has its own internal regulations to keep it at a normal set point, at its cell homeostasis. Okay, membrane carbohydrates. So attached to the plasma membrane, you have carbohydrates. And different types of carbohydrates, peptidoglycans, glycoproteins, and glycolipids. Excuse me while I get my coffee. Okay, and these proteins, these carbohydrates extend to the outside of the plasma membrane. So you can see right here, and they can be attached to proteins, but the carbohydrate itself is extending outside the plasma membrane. One of the functions is it forms what's called a sugar coat or glycocalyx. I apologize. So functions of the sugar coat: lubrication and protection for the cell, anchoring and locomotion. So these glycoproteins and proteoglycans and glycolipids help anchor the cell to other cells and also allow the cell to move if it's a cell that actually moves and is not fixed in the body. And then, very importantly, allows specificity in binding. So it allows recognition of the cell itself. And if the cell is recognized by a particular substance, solute, or receptor, then it allows that binding to occur. And then lastly, recognition. And so it allows, for example, the immune system to recognize the cell as being healthy or infected, because it will have different membrane carbohydrates displayed on the outside of the cell if there's something wrong with the cell. And then your immune system will recognize that cell as being abnormal and then destroy it. Okay, so I found a video on the plasma membrane, which kind of goes into more depth. Let me share that screen with you, if I can find it.
You can manage your team's work easily with Monday.com. Just go to Monday.com and start tracking any project or workflow in minutes. Yeah, minutes. What, you don't believe me because I'm an actor? Here, I'll show you. Get started with the...
In my second year of teaching, I was looking to improve an osmosis lab I had done the year previously. Osmosis, if you remember from our osmosis video, involves water traveling through a semi-permeable membrane, potentially a cell membrane. And all about cell transport and how materials...
Okay, so we didn't have sound. Sorry about that. We'll start over.
In my second year of teaching, I was looking to improve an osmosis lab I had done the year previously. Osmosis, if you remember from our osmosis video, involves water traveling through a semi-permeable membrane, potentially a cell membrane. And I wanted a cool way that students could model this in different scenarios. And one of my colleagues told me about this egg lab. I won't get into the whole lab though. It was actually one of the first steps of the procedure that got to me. To get ready for the lab, my colleague told me, you can soak eggs in vinegar for 24 to 48 hours and the shell comes off. Oh, so I need to make hard-boiled eggs then. No, raw eggs. But if the shell comes off, well, that's the whole point. What's underneath the shell is going to mimic a cell membrane. It's kind of modeling how a cell membrane would function, you know, if the whole egg was actually like a cell. So then you run different scenarios with it for your osmosis lab because it will be a semi-permeable membrane, like a cell membrane. I couldn't visualize this. If the shell comes off, but it's a raw egg, how does it stay together? So I have this area in my house that's designated for things for me to try out. Don't worry, I always clean up afterwards. I tried this experiment out in advance, just to be sure. The hard shell is removed, but the membrane that was always there remains. We often visualize the membrane of a cell this way, like this membrane around the chicken egg. The cell membrane is semi-permeable, meaning it lets some materials through, but not others. We have an entire video all about cell transport and how materials can pass through the membrane. A cell could never be as large as a single chicken egg, though. Why? Well, it turns out surface area is a very important thing. Remember that surface area determines the surface measurements of that cell membrane, and the cell membrane controls what goes in and out of cells. That includes food coming in, as well as molecules that are essential for metabolic processes, and then also waste going out. If volume, which is all this space inside, increases, then you will have more need for surface area, as you will have more need for food to enter, more of a need for waste to be removed, and more metabolic reactions occurring in this larger volume in the first place. If we do a little bit of math here between these two models, I'm going to use popular cube models instead of egg-shaped models because it's a little faster for me to do surface area and volume calculations. See how here there is a big difference in surface area to volume in the smaller model? 6-to-1 ratio. That means the surface area in the small model is 6 times more than the volume. Look at this beautiful ratio with so much surface area. But if we look at this bigger cube and we do some math for this model, that surface area to volume ratio decreases. Sure, the surface area is still larger than the volume in the large model, but it's only 2 times as large now, not 6 times as large. Cells are way smaller than this small model here to allow for an exceptionally large surface area to volume ratio. And a major reason why we're not going to find a cell as big as this chicken egg here. Surface area is important. And while we can model a lot of the processes of cell transport from this egg membrane and how important the membrane is, I don't want to neglect talking about the amazing cell membrane structure itself, because the cell membrane structure truly is magnificent. And since every single living thing is made up of one or more cells, which is part of the cell theory, it's a big deal because every single cell has a membrane. It doesn't matter whether you're talking about bacteria or protists or plants or animals or fungi. Even archaea aren't too cool to have a membrane. They all have a cell membrane. The structure can vary some, but we're going to be talking about some major structures of the membrane that you can actually find in most cells. We should mention that the fluid mosaic model is often what we use to describe the cell membrane. A mosaic, in case you've ever created one, we did in some of our art classes over time, arranges many small pieces together to make some large piece. You'll see that that makes sense when describing the membrane in a minute. The word fluid implies movement, and this is true for the cell membrane as the components are floating around, they're not static. So let's take a look at some of these components in the cell membrane. Looking first at a phospholipid bilayer. A phospholipid is a lipid, but an interesting one. So when you talk about a lipid in general, many lipids are nonpolar. Think of oil, for example. It's nonpolar. It won't dissolve in water. Water is polar. But a phospholipid is interesting because one part of it is polar, the head, and the other part of it is nonpolar, the tail. It's amphiphilic. Let's explain what we mean. We often refer to the polar head of the phospholipid as hydrophilic, which means that part loves water. Well, you know, if a lipid could love. The nonpolar tails are hydrophobic. They do not like water. These phospholipids arrange themselves into a phospholipid bilayer with the nonpolar areas here in between, away from any water. It also allows this area in between to be separated from the outside and inside. Water can be found on the inside and outside areas. Also, these phospholipids, they don't just stay put. They move around. It's the fluid mosaic model after all. This gives the cell membrane flexibility. Phospholipids can even flip-flop around, but that's far less common. Remember that this entire phospholipid bilayer borders the whole cell. It would be a sphere, even though we're just looking at one area of it. We have an entire video that talks about which molecules can go in and out of this membrane and which ones can't that you can view, but for now, we're going to look at some of the other structures more in depth. Cholesterol. You know, cholesterol often gets a bad reputation. And while cholesterol that builds up in the arteries can be a problem, cholesterol in your cell membrane is critical. If temperatures drop, the cholesterol can actually function kind of like spacers between these phospholipids, keeping them from becoming too packed. Or vice versa, the cholesterol can actually function to connect phospholipids to keep them from becoming too fluid in warm temperatures. Proteins. In protein synthesis, we talk about why it's so important for cells to make proteins. Many proteins are found in or on the cell membrane, and they play major roles. Peripheral proteins, like the name suggests, tend to be on the peripheral area of the membrane. So while they're on exterior areas of the membrane, they generally are not going to go through the membrane. That's for integral proteins. Integral proteins go through the membrane. Oh, and peripheral proteins can sit on them sometimes. Because of location, these proteins tend to have different functions. Integral proteins, with their potential to go through the membrane, are frequently involved in all kinds of transporting methods for all kinds of materials. Some relevance? Consider the breakfast you ate this morning. Your body digested what you ate for breakfast to obtain glucose. Once in the bloodstream, those glucose molecules can't just squeeze through the phospholipid bilayer to enter all of your cells. The glucose molecules are too big and polar. But your cells need glucose to survive, to make ATP, and they rely on integral proteins to get it. Peripheral proteins tend to be more loosely attached, since they're generally not stuck in the membrane. They have an assortment of functions, such as acting as enzymes to speed up reactions, or attaching to the cytoskeleton structures to help with cell shape. Both protein types can have carbohydrates bound to them, which can make them considered a glycoprotein. If the carbohydrates attach to a phospholipid, you could have what's called a glycolipid. Glycoproteins and glycolipids can identify the cell as belonging to the organism, self and non-self recognition, which is very important when you're fighting pathogens. They can also be involved in many kinds of cell signaling. In fact, here's some relevance. A glycoprotein known as CD4 is found on the surface of some of your immune cells. The CD4 glycoprotein is essential for some of these immune system cells to interact with each other and activate. However, it's also exploited by the HIV virus. The HIV virus uses that CD4 glycoprotein as a way to bind to helper T cells, which it then can infect. Understanding the components of the cell membrane and how those components are involved in recognition and cell signaling is critical to understanding how to fight back against many viral and bacterial diseases. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay. So that was just kind of a fun overview of the plasma membrane. Okay, then real quick, we're going to talk about three structures on the outside of the plasma membrane. The first being the cilia. And these are kind of, as it says, hair-like fibers or extensions on the outside of the plasma membrane that move, and it helps move material across the surface of the cell. So one of the places you would find cilia would be in the respiratory tract, and they beat back and forth and move the mucus up and out of the respiratory tract. You would also find them lining the fallopian tubes, and they would help move the ovulated egg towards the uterus. Okay, flagella, as we see in this picture here, usually is one, maybe two whip-like extensions on the outside of the cell that helps the cell move through the medium. So a good example would be a sperm cell, which you see pictured. And then the third extension are microvilli. And micro means little, villi meaning fingers, little fingers. And these actually work to increase the surface area of the cell and allow more absorption through the microvilli. So one of the places you would find this would be lining the digestive tract. And you have these microvilli that increase the surface area of the cell, which then increases the ability of that cell to absorb nutrients in through the digestive tract. So here's this electron micrograph of microvilli, and then here's a cross-section picture showing that microvilli. And singular is microvillus, microvilli is plural. So you need to know the difference between these extensions, what their function is, and where you would find them, okay? Here is just another illustration of cilia, and it looks like hair, kind of like a cell with hairy surface. And it's kind of like you're rowing. The hair, if you look here, try to imagine it, you have a movement that's like what we call the power stroke, and then it returns back to its original position, rows forward again, returns back, and rows again. So it's kind of like rowing with a paddle. Okay, so we are going to move on to whoops, wrong one. Diffusion and osmosis. If I can get that to open up. Okay, share that screen with you. Okay. Whoops. Try to go to the first slide. Let's try that again. Okay. Actually, before we start this, I'm going to take roll really quick. So please unmute when I call your name and let me know if you are here. Okay. Kayla.
Here.
Reagan.
Here.
Kelsey.
Here.
Tana. Tana Frederick. Stevie. Josh Kettle.
I'm here.
Thank you. Joel Langemeyer. Joel.
Here.
Thank you. Taylor Lobeda.
Here.
Taylor Lavelle.
Here.
Grace Jensen, or sorry, Mazdam Jensen. Mazdam Jensen. After all that. Grace, are you here? Andrew Marr.
Here.
Thank you. Amber Michael.
Here.
Jimmy Nguyen.
Here.
Yuen.
Here.
Marissa Pacha. Marissa, are you here? Jamie.
Here.
Thank you. And Jeanette.
Here.
Morgan.
Here.
Macy.
Here.
Ayla.
Here.
Elijah.
Here.
Araya. Araya White. Ava.
Here.
Thank you. Awesome. Okay, let me make sure I didn't miss anybody. Whoops. Oh, there's a chat. Stevie. Hannah. Okay, Stevie is here. Okay, thank you, Stevie. I got you. Okay. All right, diffusion and osmosis. Okay, so as we just talked about, the plasma membrane is a barrier. However, you need to have nutrients be able to enter the cell. Oops. Products that the cell creates and waste products need to get out. So we have the idea of permeability. Permeability determines what is able to move in and out of a cell. And a membrane that is impermeable will not let anything in or out. But that is not the case with the cell membrane. A membrane is freely permeable if it lets anything pass through it. A selectively permeable membrane restricts the movement of certain substances in and out of the cell while allowing other substances to pass freely. So it's a combination of impermeable and freely permeable. So the plasma membrane is selectively permeable, which means, as I mentioned just now, I'm sorry, more coffee. Coffee break. Okay, thank you. Okay, plasma membrane is selectively permeable, and this allows some materials to move freely across the membrane while restricting other materials, and it restricts them based on their size. So if a substance is too large, it's not going to be able to freely pass through the membrane. The smaller a substance or solute is, the easier it's able to pass through a membrane. And then electrical charge. So remember that the hydrophilic heads of the plasma membrane are water-loving, right? And water is polar. So you have an electrical charge on the inside and outside of the plasma membrane. So if a substance has a charge, it's going to be able to move across that membrane. Actually, wait, I take that back. So we'll come back to that part in just a second. But having an electrical charge determines whether or not you're able to move across that membrane, because yes, you have hydrophilic heads, but you have hydrophobic innards. And that determines whether or not something is going to be able to pass through. The shape of a molecule also determines whether or not it's going to pass through. You have those integral proteins that have receptors for certain molecules, and they have to fit into that receptor just right to be able to move across the membrane. So that's one example, the molecular shape being important. Lipid solubility goes along with the whole electrical charge idea. If it's soluble in a lipid, it's going to be able to go through, okay? Because you have that hydrophobic core that it's going to be able to dissolve through it. Okay, so transport across the plasma membrane can be either passive, which means you're not using any energy. And when we talk about energy, we're talking about ATP. Or transport can be active, which means it does require energy in the form of ATP. So, keeping that in mind, when we talk about diffusion, diffusion is a passive process. Well, there's different types of diffusion, but when we're talking about simple diffusion, that is passive. It does not require any energy. It is a random process that we'll talk about more how that happens in just a moment. Okay, so simple diffusion. Diffusion, the definition is the movement of a substance from an area of high concentration to an area of low concentration. And we often refer to this as it's moving down its concentration gradient. So let's write this down, down the concentration gradient. So understanding the concentration gradient is important. You're always moving, when we're talking about a passive process, from areas of high concentration to areas of low concentration. That is always the rule when we're talking about simple diffusion. You're moving from areas of high concentration to areas of low concentration. So in this picture here, try to imagine that we have a beaker of water and we drop in a couple drops of dye. So in this picture, the dye is green. And so the dye itself is really concentrated with pigment molecules. When you drop it in the water, those pigment molecules, they're in a high concentration. When you first drop them in, they start to spread out in the water. And they spread out, it's a random process, it's by the molecules moving. The molecules are always in motion. This is chemistry. They're always in motion and they bounce off each other and they slowly start to spread out to fill up the space that they are in. So eventually that beaker of water turns green all the way through because those dye molecules have diffused from an area of high concentration out and have eventually reached equilibrium. And so you can see that when we're talking about the dye example. So here we have an illustration that takes you step by step. So in this example, it's using a colored sugar cube and dropping it into pure water. So down here we have a low concentration gradient. Here is high, okay? So as the sugar cube dissolves, we have a high concentration of sugar right here, and those molecules start to spread out to fill up the space that they are in. And so they are spreading through the water, as you can see in this picture here, until eventually you have reached equilibrium. Which means you have eliminated the concentration gradient. And those molecules are now evenly distributed throughout the solution. So left to its own devices, when we're talking about diffusion, you have substances that move from a high concentration down to a low concentration, and left unchecked will eventually reach equilibrium, where they're equally spread out within the space that they are in. So in this example, within the beaker. Okay, factors that affect diffusion, you definitely need to know this. So the distance particles have to move. So if the particles are in a very small space, they're going to diffuse faster than if they have to diffuse across a very large space. So let's say we're talking about, trying to think of a good example. So let's say we have a small house and I sprayed air freshener in the kitchen, which is small. And if the kitchen is closed off with a door, those air freshener molecules spread out and fill up the kitchen pretty quickly. But if I open the door for the whole house, it's going to take longer for those scent molecules to spread throughout the whole house. So the distance the molecules or the particle has to move determines how quickly diffusion occurs. Okay, another factor that influences diffusion is the size of the ion or the molecule. So the smaller the molecule, the faster it's moving in space. They vibrate and move around. And so they're going to move and bounce off each other faster and diffuse faster the smaller they are. Larger molecules are moving slower and they're bouncing off each other slower and diffusing through that medium slower. So this table is really helpful. So smaller molecules, you have faster diffusion. Larger molecules, slower diffusion. Temperature affects diffusion as well. And temperature basically speeds up that random movement of molecules. And so they start moving faster and bounce off each other faster and spread apart faster. The lower the temperature, the slower they move. Okay. You have faster diffusion with higher temperature. So think about it this way. Let's say you're wanting to make pasta and you need to put salt in the water. Do you put salt in the water when it's cold, or do you put salt in the water when it's hotter? Which way is going to have that salt dissolve faster? The cold water or the hot water? Yes, Drew, that's right. Hot water. That salt will dissolve faster in hot water. So I have pots and pans that will pit if the salt sits on the bottom too long. And so I always add it when the water is hot, and then I only have to stir it for a couple seconds and it's dissolved because it's hot. If you're trying to dissolve it in cold water, you're going to be stirring for quite some time so that that salt dissolves. So that's my real life practical example of dissolving with salt in hot water. Okay. Concentration gradient will also determine how diffusion occurs, how quickly diffusion will occur. So the steeper the gradient, so if you have a super high concentration of one substance and an extremely low concentration on the other side, the diffusion is going to happen faster. Okay. If the concentration gradient is small, like the concentrations are similar, but just slightly off, diffusion is going to be slower. Electrical forces. Okay, so opposites attract, like charges will repel. So if you have two positive molecules, they're going to repel each other. If you have a positive and a negative molecule, they're going to attract to each other. And that will help determine how quickly diffusion is going to occur. So here we're looking at an illustration that kind of shows some of the substances that are going to be able to move through a membrane. So notice right here, lipid soluble molecules are able to diffuse through the membrane because the core of the plasma membrane is a lipid. If it's water soluble, it cannot pass freely through because the water can't pass through that lipid center of the plasma membrane. So in that example, you would have to have channel proteins that allow substances like water and ions that have a charge to move through the plasma membrane because they would normally be repelled by that lipid layer in the center. Then we have large molecules here. They can't pass through the lipids. They can't pass through the plasma membrane unless they have a carrier protein or a transport protein that actually allows them to pass through because they are large. Okay, so we have another video to help illustrate diffusion. And I'm sorry, it's Amoeba Sisters again, but I thought it would be helpful to have a video kind of illustrating this diffusion process. So let me share that screen with you.
It's already available in parts of select cities.
And it's rolling out in cities around the country. 25
Good for
times
you.
faster than today's 4G networks.
It's the fastest 5G in the world. This is 5G built right.
Verizon. Okay, here we go.
Have you ever heard of the ick? The disease, that is, that can afflict aquarium fish? I will never forget the first time my fish caught ick. Well, it's called ick. It stands for this. So, ick it is. It's actually caused by a protist, a parasitic one. In our ecology video, we talk about parasites and how parasites benefit while causing harm to their host. And in my case, it was all of my beloved fish. Well, okay, technically it affected Gertrude first, but then it spread because it is a contagious disease in fish. Ick typically causes these white dots. It gets on their fins, on their bodies, on their gills, all over. It can even be lethal if not treated. So it's bad news, and being a guppy enthusiast, I was really worried about all of my guppies. This is not a video about parasites. This is a video about diffusion. So you're probably wondering, how is she going to connect diffusion to this parasitic protist disease? Well, it's because of the treatment. You see, there is a commonly purchased treatment for the disease of ick. One common treatment option contains an anti-parasitic called methylene blue. Its name is not misleading. It is quite blue. The directions on the back of the medication outlined how many drops to add per gallons of water, and I watched and tried to encourage my fish to swim towards the medicine, but they were panicked, maybe because a portion of their water was changing colors, but also probably because of the kid trying to get their attention to swim towards the medicine. But you know what I didn't know then? I didn't need them to swim over there. The blue drops would spread on their own in a process known as diffusion. Diffusion is when the net movement of a substance travels down its concentration gradient. That means it moves from a high concentration to a low concentration. When the methylene blue drops were added into the water, they didn't just sit there. The molecules traveled in the water through diffusion. The high concentration of molecules, which is where they were dropped, spread to areas of low concentration in the water. Pretty soon, the water had a uniformly blue appearance as the molecules eventually are evenly dispersed. This was very important for treating my fish because the treatment reached all of them and they soon got better. Diffusion doesn't just happen in water. For example, it occurs if you spray an air freshener in the air. The net movement of molecules travels from an area of high concentration of the air freshener to a low concentration of the air freshener. Pretty soon, it's likely others would smell the air freshener, even if they were quite a distance away. Now, I want to mention two important points. First, we say net movement because that's the overall movement. But it doesn't mean that the molecules can't move around the other direction, and it doesn't mean that the molecules eventually stop moving either. The molecules are continuously moving, even when equilibrium is reached. It's just the overall movement, the net movement, is from a high concentration to a low concentration until equilibrium is reached. Second, diffusion is a passive transport. That means an input of energy is not needed. We say that passive transport does not require added energy. When we talk about diffusion of molecules in cells, for example, the diffusion of oxygen molecules into a cell, it is classified as passive transport. A concentration gradient is itself a form of potential energy. Passive transport is different from other processes that we detail in our cell transport video, like active transport, where an input of energy is required. And speaking of our cell transport video, we outlined that there is also a type of diffusion called facilitated diffusion. Facilitated diffusion is when molecules still have a net movement of a high concentration to a low concentration, but they may be too large or have other characteristics that prevent them from directly traveling across the selective cell membrane. Therefore, they have to go through a protein channel. It's still diffusion because it's still passive transport and it's still traveling down its concentration gradient. It's just that in that case, it generally has to go through a protein to get inside. Now, there are factors that can affect the rate of diffusion. We'll mention just a few of them when considering these small molecules here, diffusing in simple diffusion. Distance. The The greater the distance that needs to be traveled, the slower the diffusion rate. For example, you could observe a difference in a 5-gallon tank versus a 55-gallon one. Temperature. Okay, really quick, would you think a higher temperature or a lower temperature would increase the diffusion rate, assuming everything else is constant for the point of the comparison? Generally, a higher temperature. A higher temperature generally would mean there is more movement of the molecules, so the diffusion rate is increased. This has to do with energy, so check out the link in the description for more about that. Characteristics of the solvent. For example, is the solvent very dense? That could slow the molecules down and decrease the diffusion rate. Characteristics of the molecules, or whatever substance is traveling through diffusion, since technically not everything that travels in diffusion is defined as a molecule. What is the mass of the substance traveling? Generally, a substance with greater mass will have a lower diffusion rate when compared to a substance with less mass. Characteristics of the barrier, if diffusion is crossing a type of barrier. To explain that, let's consider diffusion across a cell, where the barrier would be the cell membrane. Small, non-polar substances pass through a cell membrane easier than something large or polar. That will affect the rate of diffusion. The surface area and thickness of the cell membrane also will alter the diffusion rate. For example, a large surface area and thin membrane would generally allow a faster diffusion rate than a smaller surface area and thick membrane. By the way, there are more factors that can affect the diffusion rate than just those 5 we listed. In fact, you may have already thought of another factor when seeing our simple diffusion image. Generally, if I were to increase the concentration of the substance here, that would cause a larger difference between these concentrations, and that would also increase the rate of diffusion. Before we end, you might wonder, why should I care about this diffusion thing? Okay, other than the aquarium fish that I love, realize that diffusion is critical for all of life. Cells are the living unit that builds up all life, and materials that cells need to survive have to get in, and waste molecules of the cell have to get out. Now, while diffusion is certainly not the only kind of cell transport, diffusion is responsible for many critical items. One example, as you sit here right now, diffusion is responsible for oxygen leaving the alveoli of your lungs to your blood. Likewise, the waste gas carbon dioxide is able to exit your blood to the alveoli, a critical process made possible by diffusion. Well, that's it for the Amoeba Sisters. And we remind you to.
Okay. I thought they had good illustrations. Let's go back to where we were. Okay, so now we're going to talk about osmosis. Osmosis is diffusion, but it's the diffusion of water across the membrane, not a substance, not a dye molecule, not sugar, not an ion like calcium or magnesium, but it's the movement of water only across a selectively permeable membrane. It's still diffusion, it's still passive, but the water molecules will move towards the area of higher concentration. So, where's my pen? We look down here. We have our selectively permeable membrane. The water molecules are the little blue molecules, and then the green would be sugar. So if we have sugar water on one side of the membrane and just water on the other, water molecules are going to move from an area of high concentration of water molecules to an area of low concentration of water molecules, okay? And it is trying to reach equilibrium again. Remember, this is still a passive process. No energy is going into osmosis. So if we're talking about the sugar concentration on the right-hand side, the higher the concentration of solutes, sugar, the lower the concentration of water. So we're talking about water concentration levels. Whoops, back up. So what is cool about osmosis, what is cool about osmosis is we have water moving from an area of high concentration of water to area of low concentration of water. And when it does that, the net volume increases and the water level will increase. And until it will continue to increase and raise up on that side, and the water level will decrease on this side until you have reached equilibrium of solute concentration. And so this is across the selectively permeable membrane, and it actually causes a physical change that you can see. So in this illustration, we have two concentrations of solution, and they're separated by a selectively permeable membrane. Whoops, oh, come on. Selectively permeable membrane right here. So we have an area of high water concentration here, an area of lower concentration of water on the right-hand side. So you end up with a net movement of water across that selectively permeable membrane. When that happens, it actually works to increase the volume of water on side B, as you can see here, until you have reached equilibrium of concentration of water on both sides. And then on this side, the water volume decreases while the water volume increases until you've reached equilibrium, and then you have equal amounts of water diffusing across both sides. And because you have equal concentrations, you have reached equilibrium, even though you have one side that's increased in volume and the other side that's decreased in volume. So it's kind of a really neat phenomenon to see. And I'm sure you're going to do a diffusion and osmosis lab if you haven't already. Real quick, have you guys done a diffusion and osmosis lab yet? Someone tell me.
No.
No? Do you know if you have one scheduled? Okay, well, hopefully you do, because it's possibly today. That's cool. Okay, so it's a neat lab. You can actually observe and see weight changes where water has actually moved across the plasma membrane or a selectively permeable membrane. So you can actually weigh the differences and see that it has occurred. So it's pretty cool. So that will help with your understanding of osmosis a lot. Okay, so we'll talk about what happens in a cell. A couple terms to know: osmolarity and tonicity. So osmolarity is the concentration of substances in a solution. Tonicity is talking about how the turgor of a cell, if it's really tight, if it's loose, if it's in a perfect happy place. So keeping that in mind, we're going to talk about three scenarios. You have three different osmolarities of solutions. So if you put a cell in an isotonic solution, which isotonic means the concentration in the cell of solutes is the same on the inside of the cell as it is on the outside of the cell. So if the concentration is the same inside and outside, then the net movement of water across that plasma membrane is going to be equal. And so you're not going to see any change in the size or shape of that cell. So isotonic, let's put this here, equal concentrations, okay? If you put a cell in a hypotonic solution, which would be this picture here, this is a hypotonic solution is where the concentration of water on the outside is much higher than the concentration inside. And so you end up with a net flow of water into the cell, which causes the cell to swell. So it's gaining water. And if the solution is very hypotonic, like let's say pure distilled water, and you put red blood cells in it, those cells will swell up and gain water until they eventually burst because they can't reach equilibrium. They have taken on so much water they can't handle it. So when they rupture, this is called hemolysis. Hemo referring to the blood cell, lysis is burst. If you put a cell in a hypertonic solution, so let's say we have blood cells again, red blood cells, and we put them in a concentration that's high in salt. So the concentration outside of water is much lower than the concentration inside. You end up with a net flow of water out of the cell, and that causes the cell to shrivel up and shrink because it's losing water. It's trying to reach equilibrium and the water is flowing out of the cell. And so when it shrivels up and shrinks, that is called crenation, which is, these are all good, important terms that you need to know. I'll ask questions in reference to probably red blood cells and what happens when they're placed in each of these types of solutions. So you need to know these terms. Okay, going through this a little more in depth. If we're talking about isotonic solutions, this is where, pardon me, okay, coffee. Okay, so in an isotonic solution, the concentration gradient is equal inside and outside. So the net movement of water across the membrane is equal. This is what it would be inside your body. This would have the normal red blood cell size and shape and allows it to function. So the fluid in our blood is isotonic to the red blood cells. The concentrations in the fluid outside and the concentrations inside the red blood cell are equal. If we put that red blood cell in a hypotonic solution, which means that the concentration of is lower on the outside of solutes, you end up with water flowing inside trying to reach equilibrium. And what happens is those water molecules flow inside the cell trying to dilute the solution essentially, and that causes the cell to swell up. And so you can see in this picture here, you have a swollen red blood cell that's round. That's not the normal red blood cell shape. And it can actually rupture or lyse, which is hemolysis. So we want to remember that term. Hemolysis. Okay. If you're putting that same red blood cell in a hypertonic, hyper usually means, you think about a kid who's hyper, they're hyperactive, they have a lot of energy. So in a hypertonic solution, you have a high concentration of solutes outside the cell. So let's say we put the cell in a high concentration of salt water. The water inside the cell is going to flow outside the cell, trying to reach equilibrium. And so the cell loses water and it shrivels up. And that is called crenation, which is an important term for you to know. Okay. So different types of transport, and this was kind of touched on in the diffusion video. Just checking our time, we got about 10 minutes left. We have carrier-mediated transport. All right, somebody's texting me from class. Okay, we'll deal with that later. Okay, carrier-mediated transport. This is where you are using proteins that will transport ions or organic substances like glucose across a plasma membrane because they cannot pass through that lipid center of the phospholipid bilayer on their own. So they need to have a protein that can transport them across the membrane so they can enter the cell. Okay, two main types of transport. For carrier-mediated transport, you have symport or co-transport. They mean the same thing. And this is when you have two substances that move in the same direction at the same time. So, for example, if a positive ion moves across the membrane, a negative ion will follow it. They're attracted to each other, so they move across the membrane or they move in the same direction together. So co-transport. Antiport or counter-transport is when you have one substance moving in while another one moves out. A good example of this would be sodium and potassium. I think that's a K, potassium. So we have what's called I'm not going to go there yet. So if you have, let's say sodium is moving in, potassium is moving out. So that's counter transport. I like these terms better. I think they're more descriptive. That's my hint to you. If I use symport or antiport, I will include the other term as well so that you are able to remember both. Carrier-mediated transport. This is called facilitated diffusion. This is a passive process, does not require energy in the form of ATP. And you have carrier proteins that will transport molecules that are too big to fit through a channel protein. So channel proteins are great for ions and water, for example, but when you have large molecules like amino acids and glucose, you need a carrier protein that will transport it through that membrane. Still passive. So what happens, let's say we're talking about glucose. Glucose binds to a receptor on the carrier protein that's embedded in the plasma membrane. This would be an integral protein. Okay, so it binds to that protein. When you have that binding occur, it causes the protein to change shape, which then allows the molecule to pass through into the cell. Even though it's changing shape, it's still passive and it does not require energy. I'm so sorry. Important to remember that the receptor site is specific for each particular molecule. So you have receptors for glucose, you have receptors for amino acids, you have receptors for certain other molecules. So they have to bind to that specific receptor that they match with in order for this process to occur. Remember, we talked about how shape determines function when we're talking about proteins. It's the same for any molecule, especially when we're talking about receptors. The receptor is specific for each individual type of molecule that needs to enter the cell. So in this example, we're looking at glucose. We have an integral protein, a carrier protein. The glucose binds to a receptor site. The fit is important, so that's how you determine it's actually the right molecule and the right integral protein. When it binds, it causes it to change shape, which basically allows that glucose molecule to enter the cell. This is passive. No ATP. Okay, I'm not going to do the video for osmosis, but I will post the videos tonight, and I will include the osmosis video for you to look at as well. Okay, talking about carrier-mediated transport. This is where it's active, that it actually requires energy. So this is active transport of substances like proteins. Oh, sorry, active transport proteins moving substances against their concentration gradient. So up until this moment, we've been talking about moving substances down their concentration gradient from areas of high concentration to areas of low concentration. With active transport, we're reversing it. We're moving substances from low concentrations up to high concentration against its concentration gradient, against the natural flow that it would normally go. This requires energy. That's why we call it active transport. So ATP. Okay. So examples of this would be ion pumps. And a really great pump that you will get to know this semester is a sodium-potassium pump, which pumps out, I think it's 2 sodium for every 3 potassium. They're antiport or counter-transport molecules, but since they're going against the concentration gradient, it requires energy in the form of ATP to do this. So we'll talk about the significance of the sodium-potassium pump later. You will definitely need to know that. Okay, exchange pumps. These are pumps that move 2 ions in the opposite direction at the same time. So the sodium-potassium pump is an exchange pump, exchanging sodium for potassium. So here's an illustration. Oh, I was wrong. 3 sodium for every 2 potassium. So this is a sodium-potassium pump. Requires energy. It pumps out sodium, so 3 sodium molecules for every 2 potassium, 3 sodium ions for every 2 potassium ions. The whole purpose of this sodium-potassium pump is to build up the ionic charge difference across the plasma membrane. So you end up with a positive charge across the plasma membrane because you have more sodium on the outside, more positive charged ions on the outside than you have on the inside. And that builds up a charge difference across the plasma membrane. That becomes really important when we talk about neurons, and neurons is the big one. So this will come back again when we talk about neuron physiology. Okay, different types of endocytosis. Endo means in, cytosis means cell. So this is when the cell is taking in substances. You have receptor-mediated endocytosis, pinocytosis, and phagocytosis. So with receptor-mediated endocytosis, you have specific receptors bound to the plasma membrane, and when the molecule that matches with that receptor binds to it, it causes the cell to basically turn in on itself and form a vesicle. And it brings it into the cell. With pinocytosis, this is often referred to as cellular drinking. This is when the cell membrane folds in on itself and takes in fluid, takes in large amounts of fluid into a vesicle, into the cell. So this is one way the cell is able to get fluid inside. And with phagocytosis, this is often referred to cell eating. This is when the cell engulfs large particles, or let's say a bacterium, because it's a white blood cell, engulfs it into a phagosome and digests it down. So things that will affect the rate of phagocytosis is how many pathogens or cellular debris are on the outside of the cell that need to be taken in. Substances that are involved include bacteria, viruses, cellular debris, dead, worn out cells that need to be recycled. Okay, This is just showing more detail of receptor-mediated endocytosis, where you have these little receptors for these molecules that bind to it, causes what's called a coated pit. It folds in on itself and forms a vesicle. That vesicle enters the cell and fuses with a lysosome, which digests those products that were brought in and then remove them. So this example is just receptor-mediated endocytosis. I'm not going to go into super detail asking questions about it, but it's just there for your information. Exocytosis. Exocytosis is when the cell is releasing substances to the outside of the cell, such as fluids or solids that the cell has created. We don't understand how the rate is affected, but substances that are involved would be fluids or cellular waste products that need to exit the cell, or secretory products that are made in the cell, like hormones that need to be released outside of the cell. Okay, we have finished that lecture. Is there any questions, because we're actually out of time right now, are there any questions about this lecture today? Okay, what is the best way to reach me if you have questions about Top Hat? If you need to reach me right away, text me. I will not be able to check my email until this evening. So if you need to reach me sooner, text me. If you're having technical problems with Top Hat, if it doesn't have to do with the due date, or hopefully you're all signed up for Top Hat now. If it's a technical issue, I would reach out to tech support for Top Hat. That's an educational program. They should have people available on a regular basis for that. But I will do my best to help you as much as I possibly can. Okay, are there other questions? Okay, did everybody hear their name called? So real quick, is Tana here? Grace? Marissa? Or Araya? Okay, awesome. Quick update. I'm planning on returning back to the classroom next week, as long as the dial does not hit red for COVID. If the dial hits red, I believe the school policy is we go online. So we will know tomorrow what happens with the dial for COVID. So if we stay in the orange, we'll go back to regular classes next week. I will send an email announcement out, letting you guys know exactly what to expect. I should know by tomorrow, and I will send an email out to everybody about that. I know some people are kind of nervous about returning back to the classroom. I get that, I am too. But we all need to wear our masks, you hand sanitize, be very careful about social distancing. I think we'll be okay. But if you have concerns, let me know. I will pass it on to the chair, Steve Bassett, and let him know, okay? All right, guys, I hope you have a fantastic day, and I will see you soon, hopefully next week. And check Canvas and your email for announcements. Have a great day.